Hello everyone, I'm Mersa Mardikor. I am from Wording Lab and I want to talk about modeling aspects of the language of life through transfer learning protein sequences. We want to predict protein structure and function, and there are different routes like combining machine learning and coevolution or using language models and creating informed embeddings. Authors talking about restrictions of evolutionary information because it needs lots of computational power, it's time consuming, and also it's not effective for small proteins that we don't have enough evolutionary information from. So that's why they are using ELMO, which is embedding from language models. We are dealing with corpus of sequences in protein databases, and we want to predict most probable next word. And in this way, we autonomously derive syntax and semantics in our hidden layers. There are two different tasks that the authors are evaluating. The one is per residue embedding, and the other is per sequence embedding for predicting protein localization and secondary structure. And then the overall workflow is that they are dealing with input sequences, they are tokenizing them, and then in the second layer in here, they are mapping them into an embedding layer. So they derive fixed continuous vectors that are independent of each other. And then in the next layer, they are using LSTM, which processes the information sequentially, and that's why we can learn the interconnectedness of amino acids toward each other. And then in the next layer, next LSTM layer, they are trying to predict the masked amino acid based on all the given amino acids before that. So let's see some results. Uh, just some notes here. The ones that have hash in front on top of their bars, it means that they use evolutionary algorithm and information. We can see that in a secondary structure prediction, a netsurf P is the best, and in localization prediction, DeepLoc is the best. So they said that their predictions wasn't was good enough, but not was but wasn't the best. The reason is that they were kind of mm, assuming different options. One would be uh, that they could use transformers in order to caption the interconnected of amino acids better. But more than that, they were talking about if we use language models, we can learn and write protein sequences, but we are missing survival of fitness by evolution. So it's a good question that how we can get the state of the art predictions without using mm, with using with being effective in computational power in time and also getting the most of the information. So this is an ongoing problem, but the just another point that is good to mention that they were talking about machine learning is not a black box, and in the right graph you can see two different scenarios when they are using trained embeddings and untrained embeddings, and they are seeing that they can capture the localization and membrane predictions of sequences when they are having informed embeddings. I'm sorry, trained embeddings. That's the kind of the total overview of the paper, but I encourage you to deep dive into this great paper for more information. Thank you so much for listening to me.